start now. And yeah, hopefully it won't take too long. It's a very easy session. Again, I apologize for any background noises. I am still on campus, that's why. You guys hear me clearly? If you don't, I can raise my voice. All right, perfect. And yeah, let's get started. So today's PAL session is about smooth muscle physiology. So let's just get started right away. Let's see. So first off, in the lecture, it discussed the sources of calcium leading to smooth muscle contraction. Now, before we do that, I would just want to revise a quick concept over here. Remember that in skeletal muscle contraction, remember skeletal muscles? In skeletal muscle contraction, right here, this requires calcium for it to occur. If you remember, you need calcium from the sarcoplasmic reticulum over here, from right here, for it to work. That's very important for us to start with. So the same thing is required in smooth muscle contraction, which is calcium. We need calcium. Now, why do we need calcium? It's because of a specific process we'll discuss in just a bit. Right now, I want to discuss where the calcium comes from. Where does it come from? How does it enter the cell to cause smooth muscle contraction? Well, first off, we have very specific sources. There are two specific sources for calcium contraction. So I summarized them over here. By the way, if I'm going too fast, if I'm going, if anything is not clear, you can just type in the chat. I'm keeping an eye on it and I'll see it in probably a few minutes, just a few seconds, whenever I glance at the chat. So yeah, so there are a few key points to understand about, what do we call it, about smooth muscles. Wait, 1 second. So the sources of calcium for smooth muscle contraction divide into two things. Number one, it's extracellular sources. Number two, it's intracellular sources. Now let's go back to the diagram really quickly so we can discuss them. So for extracellular sources, it's from two specific places. Or well, it's not two specific places, but it generally has to do with the cell membrane. Well, first off, the first source, as I've written down here, is through ligand-gated calcium channels. Your second source is through voltage-gated channels, and your third source is through stretch calcium channels. So let's take a look at them here. So first off, we said voltage-gated channels. Now, for example, say we have a neuron over here. Let me draw a quick neuron. So you have a neuron over here, innervating the cell, something like that. There are the dendrites, whatever. So this, for example, binds to the calcium, to the membrane, causes release of neurotransmitters, whatever. This causes the voltage inside the membrane over here to change. This change of voltage in the, what do we call it, in the cell membrane of the smooth muscle causes the channel to open, This allows calcium to rush into the cell. The second source could be through a ligand-gated channel. So, very similar concept. Say we have another neuron here, for example. Say a neuron. Really. What do we call it? Okay. I think the audio is working. For some reason, it told me disconnected. Anyways, so we have another neuron, for example, over here. Let's say this one over here. This releases a neurotransmitter over here, which binds to this channel, for example, and causes its opening. So it's a ligand. So this neurotransmitter that binds directly to this channel, which opens this channel, and this causes the calcium to come in. So that's the second source of calcium that could be occurring here. Let me delete this really quickly so we can see it a lot better. Someone saying anything? Okay. And finally, your third source of calcium from the extracellular is through mechanical calcium channels. What happens is, for example, say we have a big hand over here, a very big hand or something. That is the worst hand I've ever drawn in my life. Well, say we have a hand that compresses this smooth muscle. What this causes is this channel to stretch wide open. Oh, you can't hear me well? Is it better now? Is it better now? People in the back. Oh yeah, yeah. Okay. Let me try to distance myself a little bit. Do you hear me better now? Do you hear me better now? Okay, perfect. So, what do we call it? Let's see. So I mentioned that there are let me repeat this point. So I mentioned there are stretched channels over here. So these stretched channels, when the cell membrane of the muscle stretches for any reason, it could be just because of like motion. This causes this channel to open wide open and allow calcium to rush into the cell. So again, this is the third source from the extracellular environment. Okay. All right. So lastly, before we move on to the next point, is that calcium may also come from intracellular environment. Now, the way it comes from the intracellular environment is through two specific methods. Number one is through the IP3 second messenger system, and number two is through calcium-induced calcium release. Yes? Yeah, I'll recap it at the end again for you. I'll do it a bunch of times, Aslam. So let me go back here. So we've talked about these extracellular sources over here. So I can discuss right now intracellular sources. So we have a G protein over here. You see this? So what happens is that this G protein binds to a molecule, for example. Let's just say there's a small hormone over here, this hormone right here. It binds to this G protein, and this results in the whole second messenger thing. I think you guys know it. And this ends up in IP3. Now, IP3, there's a calcium channel over here. It has an IP3 receptor. This calcium channel, this IP3 binds to this IP3 channel, and this causes the release of calcium from the sarcoplasmic reticulum. If you remember, the sarcoplasmic reticulum is an intracellular store of calcium. So it's a store of calcium, which is, what do we call it, released through this IP3 receptor. Another way for this receptor to be released, another way for calcium to be released is through calcium-induced calcium release. So calcium could come from the outside, it could be anywhere, like from here, for example. This calcium comes over here, enters the cell, and causes and binds to this channel over here, which is a calcium channel, and this causes the release of this calcium over here. So these are the two methods in which calcium gets released from the extracellular environment. So sorry, the intracellular environment. So let me recap it all just really quickly. Just very quickly. So because of what do we call it? Some people are lost. So this is just from your slide over here, extracellular calcium, intracellular calcium. Let me just recap it over here. So again, you have three sources of extracellular calcium. Remember, this calcium, we need it for smooth muscle contraction. It's very important. We'll discuss it in a bit, but it needs to enter the cell. So for example, we have a cell over here. This is a cell, this is the nucleus, whatever. We need calcium to enter the cytoplasm of this cell. In order to reach the cytoplasm, we have, what do we call it? We have extracellular sources, and we have two intracellular methods or sources to release calcium. Two intracellular methods and three extracellular methods. Number one, we mentioned there are voltage-gated channels. So say this is a voltage-gated calcium channel. What happens is the membrane potential over here changes, and this causes this channel to open and allow calcium into the cell. So calcium rushes into the cell. Another source is a ligand-gated channel. For example, we have a hormone over here, whatever hormone, it doesn't matter. We have this thing. It binds to this channel and opens it up and allows calcium to enter the cell. The third way is through a stretch receptor. So for example, we press on the cell from here, we cause the cell to, get stretched out. When you stretch out the cell, this pulls this channel this way, and this allows calcium to enter. This is important in like very specific sensory receptors and other things. So what do we call it? This is a mechanical channel. And these are the extracellular sources. So remember, it's, what do we call it? It's voltage-gated channels, it's ligand-gated channels, and it's mechanical stretch receptors. For intracellular sources, It's through, what do we call it? The intracellular sources is through two specific mechanisms. Number one, we said the IP3 messenger system. And number two is through the calcium-induced calcium release. So what happens is in IP3, we have a G protein over here. So say that we have this G protein thing over here. What happens is that this G protein binds like a hormone over here, and this causes the increase of IP3. This IP3 binds to a channel over here, which causes it to, what do we call it? And this causes this channel to open up in the sarcoplasmic reticulum. Sorry, I just drew it here. This is the nucleus, but let's assume it's the sarcoplasmic reticulum. There we go. It binds to a channel over here. It opens up this channel, and this causes calcium to exit the cell. Exit the sarcoplasmic reticulum. So this is one of the sources of calcium from the inside of the cell. Remember this, this sarcoplasmic reticulum, if you remember, is very important for smooth muscle for skeletal muscle contraction, but it's also involved in smooth muscle contraction. The second way it happens is through calcium-induced calcium release. So calcium builds up inside the cell. Calcium binds to a channel on the sarcoplasmic reticulum, and this causes the exit of calcium into the cell, like this. So these are the sources of extracellular calcium. Do you guys hear me clearly? Do you guys want me to repeat anything here? Because this finishes this point. I can re-explain it, it's okay. Is everything clear?
Yeah.
Can you please repeat the receptor one? Are you talking about the IP3 one? The one with IP3? This one? Yeah. So basically, let me go back to the diagram over here. Where is it? Yeah, over here. Let me delete all this. Right here. Okay. So you see this IP3 right here? This is released through, if you remember this thing, this is phospholipase C. This basically causes, this is like a G protein coupled receptor, if you remember. What happens is a hormone or like a ligand binds to this receptor over here. This causes the increase of IP3. This IP3 will go travel as a secondary messenger. So it's a secondary messenger. Remember cyclic AMP? You remember that? You remember that guy? This guy? It's basically, this is his cousin right here. So IP3 just goes into the sarcoplasmic reticulum, binds to a calcium channel. So this is a calcium channel, and releases calcium from inside the sarcoplasmic reticulum to the outside of this, to the inside of the cytoplasm of the cell. Is that clear? I can repeat it again if you want. Yep, okay. So let's move on to the next point then. So just to summarize it again, we have extracellular sources of calcium, and we have intracellular sources of calcium. And they're through these channels right here. I've summarized them. I'll send you the PowerPoint later if you want to look at it, but it's already on your slides. Just to help you recall, in skeletal muscle, all the calcium comes from the sarcoplasmic reticulum, except like a small portion, if you remember the dihydropyridine channels, they come from there. That's not the focus of this discussion. So next is something called cross-bridge cycling. You've taken this in skeletal muscle contraction, so it's just a brief recap. So let's see. This is in skeletal muscle. I'll quickly summarize it. So remember how we've been talking about calcium for a while now. Let me go back. So we've been talking about calcium for a while. It's like, oh yeah, calcium. We need calcium for contraction. Okay, why do we need calcium? So calcium comes in and in skeletal muscle. Again, this is all in skeletal muscle. I can't move this thing out of the way. Okay, so this is skeletal muscle, just to keep it clear. This is skeletal muscle, skeletal. Just to recap, this calcium that we've been talking about for a while comes in, binds to the troponin complex over here, and this moves this molecule right here called tropomyosin out of the way to allow the myosin heads, if you remember the myosin heads from the thick filament, let me draw a better one. So remember this thing the one with the two heads right here. This allows it to bind to the actin. So this whole blue thing, these blue balls are actin. And this chain is tropomyosin. And this is the troponin complex. Usually tropomyosin prevents the binding of this head into the actin. And what happens is calcium comes in, it binds to troponin, which moves tropomyosin out of the way and allows the myosin heads to attach to the actin and then allow them to do the whole cycle thing. If you remember, like the whole goes back and then ATP, et cetera, et cetera. There's a summary slide for it in a bit. I'll go through it. So this is in skeletal muscle. I think you guys covered it yesterday and through your lecture. So let's talk about smooth muscle now. How does that happen in smooth muscle? It's kind of different. So what happens in smooth muscle is that instead of calcium binding to troponin, so we said it binds to troponin, calcium binds to a molecule known as calmodulin. So this calmodulin is like you could say it's the equivalent of troponin, but it isn't. So calcium will bind to this calmodulin, and then calmodulin, if you take a look over here, these are the calciums over here. This calmodulin goes in, finds this guy. This guy is called the myosin light chain kinase. Cool. So this is an enzyme. It's a kinase. This is an enzyme. Usually this enzyme without calmodulin is inactive. So this is inactive, usually. What happens is this calmodulin comes in, binds to this enzyme and activates it. You see how it like opens up this site? You see how it's like plugging in the site over here? It opens it up. This site over here is where the enzyme works. This is like the kinase activity, kinase. Now, if you know, kinases add a phosphate group. So they just take a phosphate and like shove it over here. That's all they do. So if you take a look, they're using ATP. So you see this ATP over here, it's going in and it's being used to phosphorylate this myosin head right here. So, and it turns into ADP. So what happens is, what do we call it? What happens is this light chain over here, this enzyme right here, activates the myosin heads. It activates the myosin head. Now, why does it activate it or how? Now, if you remember in like skeletal muscle, we said it's the tropomyosin moving out of the way. So it's the thin filament. You see this thin, you remember how the thin filament is like the actin and the tropomyosin. This whole thing is known as the thin filament. So this thing is the one being modified in skeletal muscle. This is in skeletal muscle. In smooth muscle, what's getting modified is the actual thick filament, the myosin itself. The actual myosin is being phosphorylated. The reason it's being phosphorylated is for it to be activated. So we're trying to activate these heads. We're trying to activate them for them to do like the whole, cycling process. If you remember, the whole binds to actin and does the whole cross-bridge cycling thing. So this is what happens in smooth muscle. So the only difference is the way it gets to this cycle. The actual cycle itself is the exact same between skeletal muscle and smooth muscle. The way it gets to activate the cycle is just different. This is the differences. So this modifies the thin filament, thin, in skeletal muscle. In smooth muscle, it modifies the thick filament, which is myosin right here. So let's go through the slides real quick here. So it says that in skeletal muscle, it's the thin filament that's regulated. In smooth muscle, it's called the thick filament is being regulated. How's it getting regulated? It's regulated through the myosin alteration, which occurs through the phosphorylation. And this phosphorylation happens by the whole cascade we just discussed earlier. So this whole thing happens in order to, activate the myosin head. This all happens to get this guy activated. Are there any questions? before I move on to anything else? Everything clear? Okay. So,
Can I ask you a small question?
yeah, sure.
The MLCK basically it releases ATP, and the ATP activates the myosin, or what does
No.
like?
It doesn't release the ATP. So this ATP is like on the side. So it's like it's currency. It's like money. So this is like it's money. It needs it for it needs it to get this thing activated, but it doesn't like produce ATP. ATP is already there, or ATP is already being like produced. Yeah. Okay. But it needs ATP.
So like how does it like phosphorylate it? The
No.
myosin.
So basically, you don't need to know the details. Basically, what happens is just breaks down the ATP. You see it? It's breaking it down to ATP. So what happens is it's like, if you know ATP, it's like triphosphate right here.
Yeah.
This is diphosphate. What happens is you see this, where did the phosphate, like this was 3-phosphate and this is 2-phosphate. Where did the other phosphate go? It went to right here. That's all that happened. You don't need to know how it happens. That's like a whole other degree, like for for biochemistry. Just know that it gets phosphorylated here to activate this myosin head. Okay. All right, perfect. All right, so I'll quickly recap it here again. Remember, calcium comes in. This is calcium right here. It could come in through here. It could come in through this IP3 thing over here. Remember, this is a G protein. IP3 can release this calcium to go outside, whatever. However, this calcium comes in, it comes into the cytoplasm right here. This is the outside of the cell. This is the inside of the cell. So when it comes in, what happens is it binds to calmodulin right here. When it binds to calmodulin, this calcium calmodulin complex become activated. What happens is this complex comes here, activates the MLCK. This MLCK will come in and then phosphorylate this myosin head. You see right here, the whole complex thing, this whole like concoction of enzymes is phosphorylating the myosin head in order for it to work. So all these myosin heads right here in the smooth muscle, like all the way over here, these ones require to be phosphorylated in order for them to do the whole contraction thing. So what do we call it? So yeah, you guys probably already know this. I'll just quickly cover it. Again, this is like the whole calcium calmodulin thing. It's turning on, it's causing phosphorylation of this myosin head. This is the myosin head right here, myosin head. And if you notice right here, it's phosphorylated. What happens is, like this head binds to the actin, and when it binds to the actin, it releases the ADP. When the ADP and the phosphate are released, it does the power stroke. When it does the power stroke, it becomes in a, what do we call it? Like it enters its cocked position. When it's in the cocked position, ATP binds again. When ATP binds, it releases the myosin head. This is just a quick revision. I'm not going to go over this very heavily because you've already guys covered it through the skeletal muscle contraction, but it's the exact same thing. It's literally the same thing from skeletal muscle. This is identical right here. This is just skeletal muscle down here. It's the same thing. Like they work the exact same way. The contraction is the exact same way. It's just how to get there is different. That's it. So let's see. Finally, before we move on to other topic. Now in skeletal muscle, if you remember, calcium binds to troponin, and then that moves tropomyosin out of the way, yada yada yada. You get all that. So the way to relax the muscle is through removing calcium. You just need to remove calcium, and basically troponin goes back, like gets inactivated, and then tropomyosin covers the whole active thing. This is how you relax in smooth muscle. In skeletal muscle. In smooth muscle, it's almost the same. So you need to remove the calcium first. The calcium needs to go away because the reason we need calcium to go away is to turn off this thing over here, the calmodulin. So once this calcium calmodulin thing goes away, what we need is another enzyme. And this enzyme is known as myosin phosphatase or myosin light chain phosphatase. Also known as MLCP. So this just does the exact opposite of what MLCK does. This thing dephosphorylates the myosin head. So remember how we mentioned over here, back here, where is it? So remember how back here we mentioned that this phosphorylation activates the myosin head. This, well, in order to inactivate it and to stop the smooth muscle from contracting, we need to remove this group. So there's another enzyme that works on dephosphorylating this myosin head. And yeah, there you go. This is a quick summary. So the process, this whole cycle thing, you see the cycle thing over here? It keeps going, it'll keep going around and around until this enzyme comes in and dephosphorylates this group down there. So you see this group down here? Until this thing is gone, it's gonna keep going. So this enzyme is responsible for inactivating this whole process. It's known as myosin light chain phosphatase. All right, is everything clear so far?
Yeah.
All right. Let's move on to the next topic. So there's something known as, we've been talking about contraction. So we've talked about how contraction happens and how the whole process is. So now what we need to talk about is like the properties of this contraction. So there are two types of contractions in a specific smooth muscle, something known as the phasic contraction and something known as the tonic contraction. So what happens is, in the phasic contraction, basically calcium comes in. See this calcium? It comes into the cell. It causes the activation of the smooth muscle, does all of that. And then everything goes back to normal. Like this calcium gets cleared, it gets removed, everything returns to baseline. Everything goes back to baseline. Baseline means that the contraction stops entirely. This is what phasic contraction, basically, it's just a quick phase. It's a phase. It happens quickly. In tonic contraction, on the other hand, what happens is that the contraction remains for a longer period of time. So we need the contraction to remain for a very long period of time. So there's a specific mechanism for it. I'll go over it in just a second. So the reason, like I'll try to correlate it very quickly. The reason we need tonic contraction, for example, there are blood vessels inside the body. There are blood vessels. These blood vessels need smooth muscle to be contracted all the time in order to maintain like a specific, state. We can't just have the smooth muscle contract just once and what we call it a day there. So it needs to be contracting the whole time. And in order for us to have a contraction the whole time, there's something known as the latch state right here. So what happens is, very simply, there's a myosin head. So let's draw a quick myosin head. So there's a myosin head. This myosin head has a phosphate group we mentioned. What happens is, this myosin head could be bound to actin. So this is actin. The moment it's bound to actin and this thing gets dephosphorylated, it's still stuck on actin, but it can't move. So it's known as a latch state. It latches onto the actin. The reason this happens is to prolong the contraction and to save energy. So as long as this head is attached to this actin, the contraction is maintained. So there's a maintained contraction. It's maintained. And that's why we need this latch state to maintain the contraction. The reason this is important is because there's less ATP being used. ATP is an energy source inside the cell. So there's a limited number of it. There's limited ATP. we can't just keep the cycle going. Like we need the smooth muscle to contract all the time. So it needs to be contracted all the time. But in order for it to stay contracted, if it were to work normally, it'll keep going through this cycle over and over and over and over and over and over again. And every time it goes through the cycle, it's burning 1 ATP. It's burning through 1 ATP and it's quickly consuming a lot of energy. So what we need to do is something known as a latch state. What happens is that this phosphate group over here just gets dephosphorylated while it is attached to the actin, and this maintains the contraction. I lost the slide. All right, this maintains the contraction. And that's about all. That's literally all a latch state is. This is all that latch state is. It's just to reduce the energy requirement.
Can you repeat it one last time?
Yeah, sure. So again, what do we call it? When smooth muscle needs to be made, like, for example, smooth muscle specifically needs to stay contracting for a very long period of time. There are like specific areas in the body that need to stay contracted. Don't worry about the slide. I'm just using a quick slide right here. It needs to stay contracted for a long time. Okay, so because it needs to stay contracted for a long time, it needs, like, if it were like there, one way it could stay contracting for the whole time is to just keep going through the cycle over and over and over and over and over again. The problem is when it goes through the cycle, it burns through ATP quickly. This ATP, every time it goes through the cycle, it's burning the ATP. There's only so much ATP inside the smooth muscle cell before it runs out. There's only so much. So what we need to do is slow down this entire process. We need to slow it down, but maintain the contraction. The way it happens is through this dephosphorylation of this group. What happens is this myosin head attaches to the actin. When it is attached, it gets dephosphorylated. If you remember, dephosphorylation means gets inactivated. But it's inactivated while it's attached to this actin. And that means that contraction is happening still. It's still happening, like it's still getting pulled, but it's just not cycling just as much. So basically what's going on is that it is just stuck at this stage to maintain the contraction for a long time. And this is known as the latch state. And this is for tonic contraction. This is for tonic contraction. I can repeat it again if you want.
But what was the tonic mean?
Tonic. So tonic means contracting all the time. So it's always contracting. So the opposite.
So like isotonics, for an example, like isotonic?
Yeah, like for example, you know what a tone is? What like tone? Like for example, when you say someone is toned, like they're muscular, so the muscle's always showing. So it's, for example, yeah, so that's what that means. It's just always contracting. So tonic means tonic equals always contracting.
Okay.
Always contracting. Yep. Phasic means it's just a phase. It's a quick phase. It contracts quickly and it relaxes. That's all. That's the only difference between them. Got it? Okay.
Yes.
So this is just a visual diagram. So basically over here, there's a stimulus. So calcium entered the cell really quickly. What happens is this stimulated the whole contraction. The muscle contracts. You can see the force right here. It contracted quickly. And then it went back to normal, like very quickly. It was just a quick phase. In this case over here, what do we call it? If you take a look at the tonic contraction, what happens is calcium, like there's a stimulus, but if you take a look, this stimulus is very long. Like this is the case for a tonic contraction. The stimulus just lasts a very long time. Instead of just calcium entering the cell the whole time for the entire time, what happens is it enters once, it does its whole thing. But what happens is that the smooth muscle is into its latch state. It enters the latch state. When it enters this latch state, if you take a look at the force here, the force is the same. The force is exactly the same. It hasn't changed. It's being maintained the entire time. The only difference is that it's much slower. You see the velocity right here and the this whole cycle thing. It goes back down a little bit. It's not back to baseline. This is baseline over here. If you take a look down here, this is the baseline. It's slightly higher. It's still going on, but it's a lot slower. It's much slower and it's more efficient. Like it's going a lot more slowly, but it's also maintaining the exact same force. The reason this happens is to save energy. This is literally just for saving energy. Save energy. Because if your muscles were to keep this velocity, like say this graph over here, let's see, say this graph over here, what do we call it? It stays like this the entire time, like this green one. This is the green one. I'm pointing at the green one. This thing stays up the entire time. The cell will quickly burn through ATP and run out of energy. It'll run out immediately. Run out. So the reason this goes back down in order to save energy. That's about all. So
I
over
have
here
a
is
question.
yep. What's your question?
Do skeletal muscles also have like something similar to a latch state, or it's only
No,
specific to smooth?
only specific to smooth muscle. You know why? Because skeletal muscle, for example, you have a bicep, for example. The bicep only needs to contract quickly in order for it to, what do we call it, to get the action done. And you don't need your bicep contracting all the time. So we don't need this mechanism in skeletal muscle. It just does its whole contracting and cycling thing on its own. In smooth muscle, it's required. And for example, you have a blood vessel. You have a blood vessel. This blood vessel needs to be contracting the whole time. Like its entire existence is just to contract. It's just contracting. If it were to contract like a skeletal muscle, it will quickly burn through the energy. The way it saves energy is by the latch state right here.
In the case
Okay.
that I'm holding something, like I'm constantly holding something, like if I'm holding a glass of water up the entire time,
Yep.
does it go through that cycle of phosphorylation, dephosphorylation? Does it
Like
keep going through that?
yeah, it keeps going. Like don't for example, if you were to hold a dumbbell, like say you hold a dumbbell and you're holding it out and you're contracting your arm, like eventually your arm will give out.
Yeah.
Like if you keep holding that, sure, you can keep holding it, but eventually you'll get tired and you'll release that dumbbell. What happens is you just burn through so much energy, you ran out of energy. The muscle just can't handle it anymore.
Okay. Okay. Thank
Yep.
you.
You're welcome. All right, so I think that covers the, what do we call it, the whole phasic contraction and tonic contraction thing. It's very easy. Basically, the only reason this happens is to maintain energy. That's about all. The force is the same, but the energy is less. The energy being expended is less. Someone asked a question. Smooth muscle. Yeah.
So for the example that someone said about holding a glass of water, wouldn't it be using the fast oxidative glycolytic fibers?
The fast glycolytic. Sorry, can you repeat that?
So for the example about the dumbbell, wouldn't like the skeletal muscle be using the fast oxidative glycolytic fibers?
I'm
for like-
honestly not sure about that. I haven't reviewed the different types of fibers that you like. I know what you're talking about, like the fast fibers and the slow fibers, right? That's what you're talking about. Like this one burns through the glucose quickly, and this one like does it slowly. Like I know what you're talking about, but I haven't reviewed it, so I can't really answer you. I can get back to you like later today if you want.
Okay, thank you.
Yep. All right. I guess there's no questions here. Like just to quickly, yeah.
So in the lack state, basically what happens, or like what I've understood in the lack state is that we basically release like the velocity, or like we decrease the velocity so that it doesn't burn out, or like we don't get rid of all the ATP. That's it. That's what all I need to know.
Yeah, that's pretty much it, Annie. The reason tonic contraction is like in a latch state, the reason latch state occurs is to save energy. It's maintaining, it's just, the force is same. Force is maintained despite low levels of calcium and cross-bridge cycling. This force is the same. It hasn't changed. Like you've already reached the maximum force. All you're doing is you're putting a lot less work in. Like you're putting a lot less ATP for the same amount of force. That's it.
Yeah.
Yep.
Right. And the lactic phase is always Like the smooth muscles are always in the lactic phase, right? Someone asked this.
Oh, the smooth muscles are always in the latch state. It depends on the type of smooth muscle. Some smooth muscles don't need to be in the latch state all the time. I don't know why this says latch state over here, but they don't need to be in the latch state all the time. It depends on which smooth muscle you're talking about. Okay, for example, in the GI, the smooth muscle needs to contract once, for example. It doesn't need to be in a latch state.
In the heart, for an example, oh no, the heart is cardiac.
Heart is, yeah, it's cardiac muscle.
All
For
right.
example, in blood vessels, it needs to be contracting the whole time. So it needs to be in a latch state.
Okay.
Okay.
Got it.
Okay. So this is just a quick summary. Again, this is calcium and calmodulin. What do we call it? They activate the MLCK. They activate this thing. This thing basically phosphorylates the myosin, and this causes contraction. And then in order to turn it off, we need the MLCP, or myosin light chain phosphatase, to remove the phosphate from myosin. When the phosphate is gone, it causes relaxation. Okay, and this is just another summary of what I just mentioned off your slide. Okay, so now we've been talking about like the way the muscle contracts and whatever. So now we've just been talking about the muscle contracting and relaxing. Now we need smooth muscle specifically needs to be like, just like skeletal muscle, there are levels of contraction. Like there's not just like it contracts and it reaches that state. There are like different levels of contraction. For example, it doesn't need to be contracted all the way through, and it doesn't need to be relaxed all the way through. So there's some control mechanisms for these smooth muscles. So there are four, like this is pretty long, so I've summarized it here. There are four factors that influence smooth muscle contraction. Number one is a balance between this enzyme and this enzyme. Remember, this causes contraction. This causes relaxation. Obviously, if this enzyme, the levels of this enzyme is higher than this enzyme, what do we call it? Then relaxation will be predominant. If this enzyme is higher than this enzyme, contraction will be higher. And this all depends on intracellular calcium, just how much calcium there is. The more calcium there is, the more active this thing is, this thing right here, because, you know, calmodulin is active, the thing we just discussed. And obviously, as calcium goes down, this thing goes down. So this is the first way it's controlled. This is the first factor. The second way it's controlled is through the phosphorylation of the enzyme itself. This enzyme phosphorylates the myosin head. It phosphorylates the myosin head. This enzyme can also be phosphorylated. Let me pull out the diagram here. So you see this enzyme right here, MLCK. What happens is a neurotransmitter or a hormone binds to the receptor over here. This causes cyclic AMP to rise through, you know, the G protein and then the adenyl cyclase, whatever. This cyclic AMP activates the protein kinase A, and then this thing phosphorylates the actual enzyme itself. So it phosphorylates this enzyme, this whole complex. What happens is when this enzyme is phosphorylated, it's inhibited, or it's less active with calcium and calmodulin. And this means that the contraction of the smooth muscle is less because this thing doesn't work on the myosin head. That's all. So phosphorylation of MLCK causes decreased affinity with calcium and calmodulin complex. The third way it happens is through the enhancement of the other enzyme. Yeah, is there a question?
Can you repeat it one last time, the MLCK? I'm sorry.
Yeah, I'll get back to it and once I finish this, okay?
Sorry, wallah.
Okay, yeah, no, that's fine. This is a very similar concept. That's why I'll explain it quickly. So what happens is, again, I think it's mentioned here as well. Like I'll go through this as well. So what happens is, you see this nitric oxide over here? See this nitric oxide? It comes into the cell directly. It's like lipid soluble. It just enters the cell without anything. It goes to this enzyme right here, guanylyl cyclase. Guanylyl cyclase will convert GTP into cyclic GMP. This activates this protein kinase G, and then this phosphorylates this enzyme. When this enzyme is phosphorylated, the phosphatase, if you remember, it becomes more active. It becomes much stronger and active. It increases its activity. When this thing is increased, it'll turn off the myosin light chain quicker, and it causes relaxation. Again, I'll repeat the other one. What happens is there's some neurotransmitter or hormone. It doesn't matter what it is. It binds to this receptor. This is a receptor. It's a G protein coupled receptor. What happens is adenylyl cyclase gets activated, yada, yada, yada. ATP gets converted into cyclic AMP. Cyclic AMP is a secondary messenger. If you remember, it's a second messenger. This cyclic AMP activates this enzyme, protein kinase A. Now remember, just a quick trick, any kinase, anything that has the word kinase in it is a phosphorylator. It phosphorylates. It adds a phosphate group to another molecule, phosphorylates. So protein kinase A will phosphorylate the actual enzyme. It'll phosphorylate MLCK, to make it very clear. And this turns off the enzyme. And this means when this enzyme is off, even if there is calcium, it will not activate the myosin actin light chain. And therefore, smooth muscle contraction is like inhibited. The same thing happens here through another system. All that's different is the way it happens. Like this one is a different molecule. It comes in here, activates this guy causes cyclic GMP activates this kinase. This is protein kinase G. This one phosphorylates this guy. And when this guy is phosphorylated, it's more active. Like this is like the opposite of this guy. So it's more active. When it's more active, it causes relaxation of the smooth muscle because it's dephosphorylating or removing the phosphate from the myosin head, if you remember. Is everything clear?
Yes.
All right, perfect. And the last thing is the availability of the binding site on the actin. So I think it's described a lot clearly. Basically, what happens is there's a bunch of molecules or like a bunch of signals that cause actin to be like more or less available to be interacting with the myosin, remember? So let me go back to the cycle thing over there. So let me just delete all this really quickly. So basically what happens is this is actin right here. This is actin. What happens is this site over here, like the way these two interact, might be less available for any reason. Like a molecule comes in, it causes the actin to be hidden away. For example, remember tropomyosin, something similar might happen and hides the actin. When it hides it, there's less interaction between the head and the actin itself. And that means that there's less contraction. That's about all. You don't need to know details. Just know all you need to know here. Let me go back to the slide. Okay. All you need to know, I think I left it here. Okay. All you need to know is that there's less or more binding sites on actin. For any reason, there might be like a molecule, a cascade of signals that led to that. That's it. You don't need to know the specifics here. Is everything clear so far?
Yeah.
Okay.
So basically, Miss, I just like, I'm really sorry, but like I like to explain so that to make sure I understand.
Okay.
So basically, what I've understood in branch 4 is that the PKC is basically an enzyme that makes sure the actin is very available. So it increases the availability of actin, and that's it.
PKC.
Yeah, I guess it's PKC or like basically we increase the availability of the actin.
Yeah. I mentioned that you don't need to know the specific mechanism of this. Yeah. You just need, it just makes more actin sites available. That's it.
Okay.
Okay. All right. So finally, we've been talking about calcium going away for relaxation for a while now. So how does it go away? Just 3 methods. There's a circa channel, there's this channel, and there's this channel. I'll explain on the diagram quickly. So, let me find a diagram. Did I not add it? I don't think I added it. I'll quickly explain it then. Let me draw a quick diagram then. Okay, so what happens is you have a cell over here. This is a smooth muscle cell. For example, this is the sarcoplasmic reticulum, SR. There's a specific channel on here known as circa. And this exchanges H+ for calcium. So H plus leaves the cell, the membrane, and calcium enters. So it's getting cleared from the cytoplasm. So calcium would be over here, will go in there. This is the first channel. So it gets pumped into the sarcoplasmic reticulum. The second channel is a sodium calcium antiporter on the cell membrane. Did I not add a slide here? I didn't. Oh. Basically, what happens is we have like a channel over here on the cell membrane. Let me delete all this mess. So we have a channel over here. It exchanges sodium for calcium. So sodium would come into the cell, calcium would go out of the cell. And it requires, I'm pretty sure, no, this one doesn't require ATP, but that doesn't matter. So it just exchanges it this way. This is another channel. Another way it happens is through the same, through the same mechanism I just described, but on the cell membrane. So calcium is on the inside, H plus. On the outside, it's just this comes in and this goes out. That's it. So these three channels are what are responsible for clearing the calcium out of the cell for the smooth muscle to relax. It's just these three channels. This is just pure memorization. There's nothing to understand about it. Okay. All right. I guess I'll move on to the next part. Oh, we're almost done. So
I have a question.
yeah, what's your question?
So for calcium uptake, it doesn't use the same mechanisms as the skeletal muscle, right? Because skeletal muscle uses calsequestrin, and I think the calcium ATPase pump. So does it not?
The calcium ATPase pump. By the way, this is the calcium ATPase pump. This is just another way to say it.
Oh,
this
okay.
is the calcium ATPase pump. ATPase. And for the calsequestrin thing, you just mentioned, calsequestrin is like a complex found inside the SR. It just binds to the calcium on the inside, if you remember.
Okay, so does it use
it's there.
calsequestrin? Okay, thank you.
Yeah, it's there. But I'm just saying that it gets to the sarcoplasmic reticulum through circa, and then it binds to like the calsequestrin.
So does skeletal muscle also use circa?
Yeah. Even cardiac muscle.
Thank you.
You're welcome. Okay. What do we call it? So this is just another summary, like on the slide he put there. Dr. Shams put it. These are just factors that influence the contraction of muscle. Spontaneous electrical activity on the cell membrane. You remember the voltage gated calcium channels? Those, like there could be electrical activity that allows those to open. Neurotransmitters from autonomic ending. Remember the ligand gated channels? Ligand gated calcium channels. These get modulated by these. Even hormones. Hormones can modulate them. And like besides these ligand gated channels, remember this G protein over here, the one we just discussed like a few minutes ago, this thing over here. It can, like hormones or neurotransmitters can bind to these to modify MLCK or MLCP. So it doesn't have to be just through calcium modification. So it can be through those two. So this is another, like, he's just telling you in other words. There are local factors such as whatever, pH, PO2, metabolites. These are just another way. Like, for example, there's a channel over here. There's another channel that binds, say, I don't know, adenosine or like CO2, something like that. This does the exact same thing. It goes through the exact same cycle, results in modulation or changing in one of these things. It's just methods. These are just methods in which the smooth muscle contraction is modified. Like this is just a summary of what we just talked about, but in different ways. And obviously stretch and distension. We mentioned at the beginning, there are mechanical channels. Let me find it. The very beginning, remember these? These, they can be stretched and relaxed and whatever, and these allow calcium inside the cell. Yep. I think that clears the whole calcium and smooth muscle contraction part of the lecture. Are there any questions here? I can repeat anything quickly before I move on. Okay. All right, so finally, like this is the last portion of the lecture. The arrangement of smooth muscle and skeletal muscle, just to quickly compare, if you remember in skeletal muscle, they're arranged into sarcomeres And then that shows like striation on the histology slide. Some of them, like the dark and white bands on smooth muscles, these are not visible, but they're there. It's not called a sarcomere, like the unit itself, this thing is not called a sarcomere, but it's like the exact same concept. Like remember the Z disc over here, this is the Z disc. This is the equivalent of the Z disc. This is the equivalent of the C disc. So it's the exact same thing, but it's just arranged in a different way. In skeletal muscle, if you noticed, they're arranged in like in a nice parallel way. They're all like facing the same direction. They're all on top of each other, and they're all nice and dandy. In smooth muscle, if you notice, these units are arranged chaotically, like they're diagonal, some of them are straight, some of them are down, some of them are up. And what happens is when the smooth muscle contracts, it like shrinks on itself. So like, for example, it starts like this, and then like contracts to this, for example. It contracts in all sorts of directions. That's the only difference. And because they're arranged chaotically, what do we call it? The histology just looks different. That's why they're called non-striated. Okay. Again, this is just a quick look at them. There's the myosin. This is the actin. This is the dense body, which is the equivalent of the Z disc, yada, yada, yada. Cool. Now, finally, what do we call it? Finally, I want to discuss these two curves very quickly. So if you remember the force length curve, what happens is that as the length of the muscle or the sarcomere itself increases, there's like a specific rise in the force. It's just how it is. It's just the properties of the muscle itself. Until there's an optimal length, which is right here, if you remember the optimal length. This optimal length is where the muscle works most effectively. Most effective. And what do we call it? When it works most effectively, it means it just contracts better. The reason it contracts better, if you take a look at this diagram, these myosin heads over here, this is myosin, attach to actin more. Like there's more heads attaching to the actin than compared to here. Like take a look over here. There's almost no heads over here. If you take a look at the diagram here on D, there's almost zero force. This is force over here. This is force. When they're not attached, there's no force, like at the length of the arbitrary length of 4 sarcomere micrometers at this arbitrary length, because there's no myosin that's overlapping with actin. By the way, I apologize for any background noise. And yeah, basically this optimal length in smooth muscle, the only difference is that it's much more prolonged. There's more. The reason is there's a very specific reason. The myosin itself, like this thing over here, is much longer. It's longer in smooth muscle. Longer in smooth. That means it covers more actin. Because it covers more actin, it means that the actual optimal length over here, like this thing, is much larger than what is in smooth muscle. So it covers a lot more, like there's more like overlap between the actin and the myosin. Another thing to notice is if you notice at the same at this length, the length where this force is the highest, right here. If you notice the passive force over here is less in skeletal muscle. If you take a look over here in smooth muscle, say right here, you notice that the passive force over here is much larger. Now, you know what the passive force is? The passive force is just, for example, you have like a wall over here. You have a wall and you have like a spring attached to it, a spring. If you pull the spring this way and cause it to be stretched out like this, like that, this spring itself has like a force to go back, like it wants to go back. That's why the more you stretch out the string itself, the more passive force there is. It wants to go back. The reason smooth muscle have more passive force is that there is more connective tissue. There's more connective tissue. This connective tissue acts like this wall over here. It's just more things pulling the muscle back. This is pulling the muscle back and forcing it to go back. This passive force is much greater in smooth muscle because there's more connective tissue around the smooth muscle. Skeletal muscle don't have as much connective tissue surrounding it as compared to smooth muscle, and that's why there is a higher passive force. So these are pretty much the only two differences between this curve and this curve right here. This is just much larger, and the passive force is much bigger in smooth muscles. Let me just read it out for you right here. So first off, the actual tension of the curve over here is over a great range. The reason is because the thick filament are longer than in skeletal muscle. The second thing is that the passive tension is greater at the optimal length than in skeletal muscle. Why? Because of the connective tissue. More connective tissue, more passive tension. And lastly, what do we call it? It's just saying over here that the actual force of contraction, if you take a look over here, it's about like 3.5. It's almost the same here. So it's just saying that the force of contraction of both muscles is about the same. Someone asked a question. The actual curves, okay, like, what do we call it? I'll try to explain it. I'll try my best to explain it. Basically what happens is the actual, okay, let me delete this. What happens is, let's go to this diagram over here. This is a clear diagram to help explain this curve. This is a simplified, you see this thing? This thing is a simplified version of this thing. Okay, so what happens is, this is a resting muscle. Remember, this is resting. It's not contracting yet. This resting muscle, if it's stretched out for any reason, say you just, again, let's draw this wall again with the spring attached to it. Like you see the spring over here, and then you stretch it out like this. You stretch it out. Over here, this length over here, if you take a look at the myosin heads right here, do you see any of the myosin heads attaching to the actin? Like, do you see any overlap? Are these guys touching this actin over here? No. Okay. So if you like, take a look, it says D over here. Since they're not touching it, at this length, this is a resting length. This is a resting length. At this resting length, there's no heads attached to the actin. Therefore, if the muscle were to contract, zero force would come out. There's no force, no force. Because there's no actin, like the myosin is not attaching to the actin in the first place. And this is represented on this curve over here. Like you see how if you stretch it out a lot, like right here, you see if you stretch it out to length 10, if you stretch it out a lot, what do we call it? If you stretched it out a lot, the force, if it were to contract, is zero. Take a look at the curve right here. It's zero. The same thing happens if you go the other way around. If you shorten it way too much, you see over here, if you shorten it way too much, this actin from this side goes over the actin of the other side. And what happens is these heads can no longer work. So the other way, it's true the other way. So there is a specific length at around this length between B and C, where the heads are attached to the actin most optimally. Most optimally. And this length is known as the optimal length. And this optimal length is where the contraction, the active contraction, is highest. That's why muscles prefer to be at this length before they contract. This is all before they contract, before contraction. This is the resting length before contract. This is resting length. Resting. What happens is when you stretch out the muscle, when you stretch it, it can't contract anymore. So it can't generate the force because the actin, as we mentioned over here, the actin and the myosin are no longer binding. But what happens, like, for example, think of the muscle like a spring. What happens when you stretch it out? Like you see, when you stretch it out like this, if you let it go, it's going to go back like this. So this is what the passive force is. It's just going to go back. It's not useful. This is not useful for contraction, but it happens naturally. Okay. Yeah, active is useful because it's like what you need. Like the muscle contracts for it to work. This is not very useful because it's like a spring. When you pull the spring this way, it wants to go back. That's it. That's all that means. Okay. Okay. Yeah, basically remember the differences. Like what I would do here is just memorize the differences between these two curves. Just remember that the myosin here is longer in smooth muscle. That's why the curve is much bigger than this. It's much wider. Like the actual curve for the active force is much wider than the one for the skeletal muscle. And remember that there's increased passive force for smooth muscle because there's more connective tissue. There's more connective tissue. I highlighted it here as well, I think. There's more connective tissue, so there's more passive tension or passive force. Tension means force, also equals force. And they both have the same amount of force generated. So these are the three properties. Just know them. If you don't understand it, just the least you should know is just remember these three properties. This is just to demonstrate that in smooth muscle, the actual myosin itself is much longer. You see the length over here? It's much longer than the smooth muscle over here. That's it. Okay. Lastly, this is the last thing I'll discuss. No, actually, there's one more thing right after. We'll finish it quickly, though. The last thing I want to discuss is this force velocity curve. So say, for example, you have like a bicep over here, okay? You have like your hand over here, you have a hand right here, and it's holding like a weight. So there's a weight over here, weight. The heavier this weight is, the less, like if you try to contract this muscle, that your hand would lift a lot slower. Than the lighter the weight is, your hand, when it contracts, it can go a lot faster into that state. This is exactly what this means. So you see the force right here? This is the same thing as load. So as the weight of this object goes up, the velocity over here of the skeletal muscle goes down. Obviously, if there's no weight, like it's just doing its own thing without a weight, it's going to be the fastest. It'll contract very quickly. This is all this curve means. And it makes sense. The only difference between smooth muscle and skeletal muscle is that at any given force, at any of these, say we pick one, at one, skeletal muscle will always be faster than smooth muscle. Even the maximum smooth muscle can do is much slower than the maximum, what do we call it, a skeletal muscle can do. It's just because, if you remember, in smooth muscle, it needs to conserve energy. Conserve energy. And there's a latch state, if you remember. Remember the latch state? The latch state? Latch state means that the contraction happens a lot more slowly to conserve this energy. Like when it's in skeletal muscle, because it happens so quickly, it burns a lot more energy. There's more ATP consumed. More ATP consumed. In smooth muscle, the reason it happens a lot slower is because of this conservation of energy. So yeah, smooth muscle remain attached to actin longer during each cycle, so less ATP is consumed. This is very
So I
important.
want to make sure that I understand because
Yeah.
our smooth muscle is in a lactic phase most of the time. That's why the velocity is very slow. That's why even if we have exert very much, a lot of force, it's going to still be slow in the
Yep.
velocity, right?
Yeah.
To save ATP.
Yeah, basically. In skeletal muscle, you want it to go fast, basically. Say like
Yeah.
a tiger is approaching you and you need to run quickly. So this thing needs to be fast.
Uh-huh.
This doesn't need to be fast. Like it can work at its own pace. And there's a latch. The reason this happens is because of the latch state.
Yeah.
Latch. Okay?
No.
Just remember that smooth muscle is a lot slower than skeletal muscle when it comes to velocity. This is all that this is all that's this saying. Does anyone want me to re-explain that? It's okay. Because we're almost done. There's just one last small point at the end. Okay. All right. So lastly, I want to make it very clear before like I explain this point. I accidentally zoomed in. Okay, so I want to make it very clear. This is different from the thing we discussed earlier. You see this terminology over here, it says tonic and phasic. This is different from this tonic and phasic. Where is it? This is different than that. This is for one smooth muscle. This is in one smooth muscle. This happens in one smooth muscle. So it could happen this, or it could happen this. In this at the end over here, like these tonic and smooth muscles, we're talking about the whole group. We're talking about multiple smooth muscles. So what happens is that some smooth muscles are considered to be phasic, like they're arranged in a phasic manner, and some are arranged in a tonic manner. What this means, literally all this means is that there are gap junctions, and in this case, there are no gap junctions. This is the only difference between these two. This means when one of them gets activated, everything else gets activated. This is all this means. So they work as a single unit, and it's like one contraction. Okay. In here, there are multiple units. So there's like 3 different neurons over here. They all like influence the contraction of each different muscle on its own. So each muscle here works independently. So the contraction of this smooth muscle doesn't depend on this smooth muscle. So they all work differently. Now, the reason we need this in the body is because some things need to be coordinated. So some contractions need to be coordinated, and they need to be very strong. They need to be strong, coordinated contractions. Here, some contractions don't need to be coordinated, but they need to be precise and accurate. And for this precision to happen, like only there's only a small amount of muscles that need to be activated at a specific time. Yeah, I will show you an example right now. Let's see. So this is again, so phasic smooth muscles work as a unit, tonic smooth muscles work as multi-unit. This is different from the phasic and tonic we discussed earlier. This is something entirely different. This is how multiple smooth muscle cells are organized. That's all. So for phasic smooth muscle, the ones that are single unit, in the GI tract over here, what we need are strong contractions. We need very strong contractions to move food down the GI tract. We don't need precision. It doesn't need to be accurate. It just needs to contract very hardly. So the smooth muscle are arranged into a phasic manner. Same thing in blood vessels. In blood vessels, we don't need like half these smooth muscles to turn on and off. All we need is all of them to work at the same time. So again, these are single unit. So this is in blood vessels, and this is in the GI. A different thing in tonic smooth muscles, like multi-unit, some examples include goosebumps, and the eye itself. So in the eye, there are muscles that are arranged this way. You see the iris itself. These muscles need to be very accurate because they allow light into they allow light into the eye itself. So it needs to be controlled very precisely. And that's why these smooth muscles are arranged into a multi-unit. These are just examples. I don't think they're coming in the exam or anything. I got them from like the internet. It's just to correlate the smooth muscles to their actions, like the actual organization. So remember, phasic smooth muscles are a single unit. Tonic smooth muscles are individual myocytes that are isolated from the neighboring cell. So what do we call it? In the eye lens, it's present in the eye to help control the shape of the eye and the way the actual light comes in. That's the only difference between these two types of smooth muscle. Is that clear? Okay.
Yes.
Okay. Yeah, that's it. Can you? Okay, cool. Yeah, that's it.
Thank you.
The lecture is very easy. Inshallah, you guys will do well. I'm praying for you guys tomorrow. I think your exam's tomorrow, right?
Yes, tomorrow.
Inshallah, you'll do well. Inshallah, it'll be very easy. If you need any help or you need any advice, I left my email at the beginning of these slides. You can email me there. You can also ask for my number if you need anything or have any questions. I can answer them. Other than that, yeah, that's it. If anyone wants me to explain anything real quick, I can do that as well. And yeah, thank you guys for attending.